Hello and a very good day everyone. I am Nursaffa Alisya from University Putra Malaysia who will be presenting my research project on the title of a new pharmacology-based investigation on the bioactive compounds and molecular mechanism of Moringa oleifera against pancreatic cancer. Pancreatic cancer is usually associated with a very poor prognosis. The 5-year relative survival rate for pancreatic cancer has shown a minimal improvement from 6% to 10% in 2014 to 2021. Despite the gradual increment, the number of deaths in pancreatic cancer is still increasing since the majority of the patients are diagnosed at the advanced stage. Moringa oleifera, which commonly found in India, Africa, and Southeast Asia, has been widely used as traditional herbal medicine in both traditional medical systems, including Ayurveda and traditional Chinese medicine. Interestingly, the in vitro study of Moringa has revealed 98% reduction of pancreatic cancer cell survival. This present study aims to evaluate the potential effect of Moringa against pancreatic cancer through pharmacology. For methodology, the study was divided into 3 steps, which are target identification, screening potential target, and node analysis. In the first step of target identification, the identification of Moringa compound were performed by using TSM-SP and TSM-ID databases. However, its constituent cannot be found in the database. Alternatively, studies related to Moringa were searched through PubMed to determine the biochemical compounds, and the compounds were taken from Zhong et al. 2020 study. As the compounds will be orally administered, they need to undergo the ADME screening to predict the oral bioavailability, drug likeness, and CACO2 permeability. The bioactive compounds that meet the requirement will then be predicted the target genes using drug bank, DGIDB, and CTD databases. Initial screening of the target genes were also conducted for the remaining compounds that cannot be found in the TSM-SP, and the bioavailability was searched through PubMed. For pancreatic cancer, OMIM and Malakat databases will be used to collect the target genes. The result for the target genes will then be presented by the Venn diagram to determine the pancreatic-related target genes of Moringa oleifera. In the final step, the pancreatic-related target genes of Moringa will be imported into string database to study the protein-protein interaction. Then, the interaction of the protein will be analyzed by determining the gene ontology and pathway analysis using the database and constructing compound target pathway network using cytoscape. As a result, there are 32 bioactive compounds in Moringa that were listed in found study. However, only 4 compounds, which are catechin, epicatechin, quercetin, and camphorol, were selected as they met the requirement of the drug screening. For the screening of the remaining Moringa compounds, only 3 out of the 25 compounds were found. Glucomoringin and glucoraphanin were reported to have a good bioavailability, while the solubility of moringinin can be improved by the formation of salts. Next, Figure 1 shows the result of pancreatic-related target genes of Moringa and its protein-protein interaction network. There are 81 nodes that overlap between Moringa and pancreatic cancer, and 373 edges where the nodes represent the target genes, while the edges represent the interaction between them. This network showed that these target genes have complex interaction between each other. From the PPI network, 9 hub genes were identified based on degree, closeness, and betweenness value, which are listed in Table 2, including TP53, AKT1, VEGFA, MAPK3, IL6, Jun, MAPK1, CCND1, and TNF. Hub gene is defined as node with high connectivity, which considered to play a crucial role within the network. Next is the result of gene ontology and pathway analysis. The result demonstrated that the most enriched biological process was associated with the regulation of apoptotic process. It has been reported that positive and negative regulation of apoptotic process are simultaneously activated through noxious and protective signal via various pathways. Therefore, it is pertinent to finely balance the regulation of the 2 signals to increase the effectiveness of Moringa against pancreatic cancer. In the CAP pathway, the enrichment pathway were mainly associated with cancer-related disease, including pancreatic cancer. Last but not least is compound target pathway network, which is constructed to show the interaction between the compounds, pathway, and the target genes. This network contains 98 nodes, which comprise of 7 compounds, 81 target genes, and 10 pathways with 1,623 edges. From the compound target pathway network, 2 hub compounds, which are catechin and camphorol, with the average node degree more than 30.909, were identified as potent compounds that are responsible for the therapeutic effect of Moringa against pancreatic cancer. Catechin and camphorol bioactive compounds found in this study are fibrin derivative, which possess the health-promoting properties, including anti-cancer. This compound play a role in the regulation of various cancer cell activities by inducing the apoptosis effect. Hub genes, including TP53, AKT1, VEGFA, MAPK3, IL6, Jun, MAPK1, CCND1, and TNF, play a crucial role within the network with high biological importance and significantly enriched in multiple pathways. Mutant TP53 has been demonstrated to induce gemcitabine resistance towards pancreatic cancer cells. Moringa could be the new therapy against pancreatic cancer since it is targeting TP53, which is the common mutated tumor suppressor gene in pancreatic cancer. In conclusion, this in silico study has revealed the multi-compounds, multi-targets, and multi-pathway, and provide a theoretical insight on Moringa intervention in pancreatic cancer, which suggests direction for further experimental research and subsequent clinical application. These are the reference. With this, I would like to thank all of you for giving your attention and listening to my presentation. Thank you.